All right, so what you want to do first, so typically like when I draw a cartoon of a neuron, you don't have to draw this part, but here we have the cell. Oh wow, I have to turn that part off. But what we have here is like the cell body of a neuron. Here I have a nucleus, and we have something called an axon. And then it ends in these things called axon terminals. So when something terminates, it ends, right? So now we're going to draw an axon terminal like this. So here's the end of an axon of a neuron. So this entire thing is called an axon terminal. Okay. Now let's draw a muscle cell. So we're drawing a very close up of the interaction in a motor unit. Remember, motor units are brains and brawn. It's a neuron, motor neuron plus muscle cell. So now we have to draw the muscle cell. And here's them, like I'm just drawing a generic muscle cell. And then draw a little divot like this. I'm exaggerating it, but it becomes important later. Okay, so this is our muscle cell, myofiber, and all, all the other things like muscle fiber, myocyte, yeah, all the same thing. Okay, so this is currently it's at rest. So what happens is that at rest, in these axon terminals, you have these little bubbles, and these are called synaptic vesicles. So these are like little pockets of cell membrane, and within the ones in the motor unit, in the motor neuron, what you have are all these, you have this chemical stored in. So I'm going to draw these as little green dots. So you have a special type of chemical, and this chemical is a neurotransmitter. It's something that transmits signals from neurons to other cells. So this neuron, it has a special name. And let me erase this part right here. So this chemical I drew right here is something called, yes, the abbreviation is ACH, but you should know the full term is called acetylcholine. So that's ACH is just the abbreviation for acetylcholine. Okay, so this muscle cell is at rest. So nothing is being contracted, and there's no activation of that motor neuron. All right, so then what's going to happen? Well, I'm leaving out part of the earlier story. So the thing is that neurons fire off these signals that involve ion movements and electrochemistry called action potentials. Do not go too far. We will get to that later on in the semester, but I'm leaving action potentials for exam 3. It's a heavy topic. Don't worry. Just know for now, there are electrical signals caused by the movement of ions. So these neurons, they fire off action potentials. Eventually, they reach the axon terminals. So now I'm showing these things called action potentials reaching these axon terminals. So what happens here is that they open these special channels. So remember I talked about ion channels? Well, what they do is open these special channels. So once you have this, it's kind of like a electronically activated gate. These ion channels are closed when there's no action potentials, but once these action potentials reach the axon terminal, they open these gates over here. Hopefully I can erase just this part. Now, this is a special type of ion channel. This is an ion channel that allows calcium to flow. And things that, remember, this is why I bring up that previous slide. There's more, in a cell at rest, there's more calcium on the outside of a cell than on the inside. So remember, things go from high concentrations to low concentrations. So actually, to exaggerate things, I'll put like, this has a very high calcium concentration. Not so much calcium inside. So remember, in terms of diffusion, things go from high concentrations to low concentrations. So calcium will flow in. Why is that a big deal? Well, with all this calcium rushing in through these channels, into the axon terminal. This causes these signals, these synaptic vesicles, to do something. This actually causes them to fuse with the outer cell membrane. So these vesicles are now going to go here, and they're going to merge with the outer plasma membrane of a neuron. So if we draw something, I'm going to show that fusion. So yeah, that's why I like to think of synaptic vesicles as like little bubbles. And now these bubbles are rising to the surface of a neuron, outer surface of a neuron, and popping. And when bubbles pop, they release their inner contents, right? So that's what we're going to do here. All this acetylcholine is now going to be released in the synapse. And the synapse refers to this overall space between the neuron and this muscle cell. So now you have all this acetylcholine being released onto a muscle cell. And this is very important because, again, this is how you get the signals from a neuron to a muscle cell. That's why acetylcholine is called a neurotransmitter. It carries that signal from a neuron to, in this case, a muscle cell. Now, on the muscle cell, you have something called sodium channels, sodium ion channels. Thing is that when there's no acetylcholine, these channels are closed. But when you have acetylcholine, these channels actually are kind of like a locked gate, and acetylcholine is the key. So when you have acetylcholine binding to one of these, let me just do this. So yeah, without acetylcholine, these ion channels, these are sodium ion channels. These are closed, but if you have a sodium ion channel and then acetylcholine binds to it, it kind of opens up these channels. So when you have acetylcholine flooding the junction between a muscle cell and a neuron and a motor neuron, these ion channels open. So now we have all these channels that can allow sodium to go through. Now, where do you typically have more sodium? On the outside of a cell or on the inside of a cell? Typically, again, remember, that's where that previous figure comes in real handy. At rest, a cell has more sodium concentration on the outside than on the inside. So all this sodium is now going to rush into the muscle cell. Now, why is this important? Well, when we talk about action potentials later on, we'll figure out, you'll know more in detail why sodium triggers something. But what sodium does is now it causes these action potentials in a muscle cell. So again, action potentials at this point in the semester, just know it's some sort of electrical activity caused by the movement of ions and chemicals. So you have this electrical activity called action potentials, and they travel across that sarcolemma. And remember, a sarcolemma is just a fancy term for the plasma membrane of a muscle cell. And this is why I had you draw that little divot right here. Not only does it travel across the length of a muscle fiber and the sarcolemma, it also travels down these things called Ttubules. So the thing is that you'll actually have multiple Ttubules along the length. So the thing is that they help to carry that electrical signals from the outside of a muscle and periphery of a muscle cell toward the innermost areas of a muscle cell. So they're kind of like, yeah, there's more than just one Ttubule per muscle cell, but I'm just showing you only one for simplicity's sake. Basically what they are is kind of carrying the action potentials and electrical activity from the outside of a muscle cell more toward the inner side parts of a muscle cell. Now, thing is that you have something called a sarcoplasmic reticulum, also abbreviated as simply just SR. So again, this is the the endoplasmic reticulum of a muscle cell. And heart cell, like cardiac muscle cells, these also have sarcoplasmic reticula as well. Same with like smooth muscle cells. But what makes this different or what makes this a little more special than a typical endoplasmic reticulum in any other sort of non-muscle cell? Well, the thing is that at rest, sarcoplasmic reticulum contains a lot of calcium. There is so much calcium up in here. Calcium, calcium, calcium. And this is a sarcoplasmic reticulum at rest. I'm showing here on the right. Thing is that you also have these calcium channels, but in a cell at rest, these channels are closed. Now, what happens when these action potentials come here? Well, the thing is that these action potentials actually send a signal to calcium channels on the sarcoplasmic reticulum. So the sarcoplasmic reticulum is kind of like a big bank of calcium. There's so much calcium held in there, but once you open this channel, remember, you have tons and tons of calcium, so much calcium that it starts flooding out. So the thing is that the sarcoplasmic reticulum is a membranous structure. Initially, this calcium is not part of the cytoplasm, or sarcoplasm in this case. But once you open these calcium ion channels, and this is due to those action potentials, this is actually the trigger that opens these channels. All the calcium floods into the sarcoplasm. So calcium increasing in the sarcoplasm. Actually, should put up here. So increase in sarcoplasmic calcium. And why do I do that whole drawing exercise? I think it's a lot easier to do. Like, this is a complex, this is why I'm trying to break it down step by step by step, so that it's a lot to take in from just one figure, but all of these kind of follow a sequence, and each of this makes a sequence in order, and it also tells you, like, okay, ions important, and things like acetylcholine are important, and also calcium is important as well. That's why if you have like too much or too little calcium, too much or too little sodium, this throws this whole system off. That's why I bring up a lot of this chemistry. All right, so then let's see this in animation form. So to recap what we just did, so I'm going to show you just an animation. This is what we call the neuromuscular junction. So here's the skeletal muscle over here. Here's the end of an axon. We call the axon terminal. So this overall interface between a motor neuron and a muscle cell is called the neuromuscular junction. So remember, you have these things called action potentials traveling along neurons. Eventually, they reach axon terminals. These open calcium channels. And remember, there's more calcium outside of a cell than inside at rest. So once you have all this calcium channels open, this allows calcium to flow into the axon terminal. This triggers the release of acetylcholine from these little membrane bubbles we call synaptic vesicles. So this causes these synaptic vesicles to merge with the surface of a neuron, releasing their contents between the muscle cell, or releasing their contents onto the muscle cell. This acetylcholine binds to these ion channels that open in response, and these ion channels are sodium ion channels. This allows sodium in. This sodium triggers another wave of action potentials, but this time along the length of a muscle fiber. And now they travel along the sarcolemma, again, the plasma membrane of a muscle cell, also within these two tubules, which are just indentations in that plasma membrane. These electrical action potentials open these calcium channels on the sarcoplasmic reticulum, causing all this calcium to flood out and into the sarcoplasm. Yeah, so that's for that previous few things in a nutshell. So the thing is that this is one of the big takeaways. Ion movement is needed to drive skeletal muscle contraction. If you have imbalances in the levels of sodium, calcium, and especially sodium and calcium, and even potassium in your muscle, anywhere in your body, this whole system breaks down.